Good morning, my dear students. Today we will discuss one more topic in third unit. The topic is transcription or synthesis of RNA. What is our last topic? Replication or synthesis of DNA. What is the replication? Synthesis of DNA from DNA is known as replication. So the next one topic is a transcription. Transcription or synthesis of RNA. So the RNA synthesized from the DNA. DNA is converts to RNA. DNA is converts to RNA is known as transcription or synthesis of RNA. What is the transcription? The DNA is converts to RNA is known as transcription, or synthesis of RNA from the DNA is known as transcription. So what is our last topic? Synthesis of DNA to DNA. Synthesis of DNA from DNA is known as replication. This is called as replication. This is called as transcription. In here, the synthesis of DNA from DNA easily synthesized. Why? Because of the DNA is having the double helical structure and the daughter DNA is also having the double helical structure, but RNA it only single helical structure. So what is the process of the transcription or synthesis of RNA? In the transcription or synthesis of RNA, involved in the three stages are involved here. So what is our first stage? First stage is the initiation, second stage is the elongation, third stage is known as the termination. The three stages are involved in the synthesis of RNA or transcription. So in here how to produce the double helical structure is converted to single helical structure. So the single helical structure, the single helical structure produced from the double helical structure, or the RNA synthesized from the DNA depends upon the one enzyme. That enzyme is called as a polymerase enzyme. That enzyme is called as polymerase enzyme. What is the polymerase enzyme structure? 4 hydrogen structure with the sigma bond. This is called as the polymerase enzyme. So the polymerase enzyme involved in the synthesis of RNA depends upon the polymerase enzyme. So we are getting the RNA from the DNA. So in here three stages are involved here. What is the first stage? Initiation. Second stage is the elongation. Third stage is known as the termination. So in initiation process, what is the RNA structure? The synthesis of RNA from the DNA. What is the RNA structure? Single helical structure. What is the DNA structure? Double helical structure. So this is called as a DNA. This is called 5 and 2 3 and 3 and 2 5 and direction is known as a this structure is known as DNA. The DNA is converts to what is the end structure? RNA, single helical structure. How to produce the RNA from the DNA? So the DNA having the three sites. One is the promoter site, transcription site and termination site. These three sites are involved in the synthesis of RNA. What is the three sites? Promoter site, transcription site, termination site. In here, each and every molecule having the three sites: one promoter site, transcription site, termination site. Promoter, transcription, termination. Promoter, transcription, termination. Promoter, transcription, termination. Each and every DNA molecule having the three sites. What is that three sites? Promoter site, transcription site, termination site. What is the RNA steps? What is the RNA synthesis of RNA stages? Initiation, elongation, termination. So in the RNA synthesis, which type of enzyme is involved here? Polymerase enzyme. The polymerase enzyme is involved in the synthesis of RNA. This is the outline of the synthesis of RNA. So synthesis of RNA from the DNA. So first, the promoter site. In initiation process, the promoter site starts the RNA synthesis. So the promoter site having the RNA polymerase. The promoter site, first the RNA polymerase having the RNA polymerase is presented at the promoter site. This is the promoter site. The promoter site having the polymerase enzyme. The RNA polymerase enzyme. The RNA polymerase enzyme is at the initiation step. It is this polymerase enzyme present at the promoter site. The promoter site, the RNA polymerase having the 4 hydrogen bonds with the sigma bond. 4 hydrogen bonds with the sigma bond. Then entering into the sigma bond in initiation process, the RNA polymerase is involved here. The elimination of the 4 hydrogen bonds is converted to the entering into the elongation step. The 4 hydrogen bonds is presented at the middle point transcription unit. The promoter site having the RNA polymerase, in this RNA polymerase it is transferred into a transcription unit. It's showing the bubble at the transcription unit. What is the initiation process? In initiation process, the RNA polymerase enzyme attached at the promoter site. It is initiation of the synthesis of RNA by the elimination of the sigma bond. So this starting point is this point is known as the initiation process. In initiation process, RNA polymerase attached at the promoter site and begin the synthesis of RNA. Then this RNA, the initiation process is over. Then after initiation process, the promoter site is converted to the promoter site is over. The next one, the transcription unit having the only 4 hydrogen bonds of the polymerase enzyme. So entering into the elongation step. In elongation, what is the elongation? Elongation means continuation process. The RNA polymerase starts the activity in the DNA middle unit. That unit is known as a transcription unit. Transcription site. In this transcription site, the polymerase enzyme react with the DNA molecule, indirectly react with the genetic information. The double helical structure, it produces the one tail-like structure. The tail-like structure having only 5-end direction only, 3-end to 5-end direction only. 3-end is here, 5-end direction is produce a small tail-like structure, depends upon the polymerase enzyme, and continuation of the elongation, the bubble from the 5-end to 3-end direction here. At the near the 5-end direction, the bubble is transferred to the near the 3-end direction here. In this elongation process, this bubble is moved from promoter site to termination site and produces a long tail-like structure. It produces small tail-like structure is converts to long tail-like structure. In here 3-end to 5-end direction is produced, long tail-like structure is produces. Then in this elongation process is over. It produces a complete RNA is produced in the transcription unit. So up to transcription, from the promoter site to up to transcription process is known as elongation. In initiation process only presented at the promoter site. The next one, elongation process presented at the only transcription site. So after transcription site or after elongation, the elimination or separation of a complete RNA from the DNA, that step is known as the termination step. In this termination means separation or isolation. So in this termination is divided into two types. One is a dependent row factor, depends upon the row factor. One is the depends upon the row factor or independent row factor. The termination is divided into two categories. What is that? That depend row factor. Second one is the independent row factor. Dependent row factor and independent row factor. This symbol is known as the row factor symbol. Symbol is known as row factor symbol. Dependent row factor, independent row factor. What is the importance of the row factor? The row factor is involved in the termination site. Termination site. These are react with the only DNA, not to the polymerase enzyme. The row factor only react with the DNA template strand only. do not react with the polymerase enzyme. So the rho factor is that the elimination of the RNA from the DNA, RNA from the DNA. What is the rho factor here? The specific the rho factor is nothing, a specific type of protein. What is the rho factor? It is a specific type of protein. Though these are the rho factor, this binds to the growing of RNA. The rho factor is a bind to the growing of RNA, and it is a termination, it is a separation from the DNA. That is nothing like the dependent rho factor. What is the independent rho factor? The independent rho factor is a termination of the in case of is brought about by termination of the hairpin structure. The independent rho factor like a hairpin, U-like structure or hairpin structure. The hairpin structure is the folded to the RNA. In independent rho factor is involved in the termination site. It produces the folded like RNA is produces, like this one, folded like RNA is produces. In case of dependent RNA is presented in the termination site, it produces the only a straight line like RNA is produces. Why? Because of dependent RNA only directly termination of the RNA in the DNA at the transcription unit. But independent rho factor, this structure like a hairpin structure or U-like structure, it is the folding of the RNA, and the next one termination is produces. In after termination, the four hydrogen bonds of polymerase enzyme is entering into the again initiation process, bind with the sigma bond and entering into the elongation process, and continuously the cycle is running in our each and every cell. This is the transcription or synthesis of RNA. What is the transcription? Synthesis of RNA from the DNA. Synthesis of single helic from the double helical of DNA. So the DNA having the three sites: promoter site, transcription site, termination site. In promoter site, bind with the polymerase enzyme. In transcription process, it produces the RNA. In termination, after producing of RNA, is separation from the DNA. So the synthesis of RNA involved in the three stages. What is the initiation, elongation, termination? In initiation, the DNA bind with the RNA polymerase enzyme. In elongation, it produces a long chain, a complete RNA is produced from the DNA at the transcription unit. After elongation, the termination stage, in termination depends upon the rho factor, or dependent rho factor or independent rho factor. So dependent rho factor, it produces a straight line like structure of RNA. A folded like structure depends upon the independent rho factor. This is about transcription or synthesis of RNA.